So coding regions can be separated from the non-coding regions or the introns. Coding regions in the genes are obviously the exons. So here we mean the exons, and then we have exons, and then they are separated by introns. And introns, they start with GT. We call it as donor site, and acceptor site is where the introns, they are ending with the sequence like YAG. Y can be any pyrimidines. Mostly we put AG as the ending sequence. So GT, AG, these are donor and acceptor sites. These are the canonical forms. So over 99.24% we will observe these nucleotides. There are some non-canonical forms also in addition to this. So like TSS, the flanks of splice junctions, they also show bias distributions of nucleotides in certain positions. So these bias distributions can be used to predict those splice junctions. That's what we also did a similar activity in identifying the TSS. So we can do something similar here. Here is the example. We can see one more time. We have exons, and then we have donor site where we see a big GT. That representation, guys, is called as sequenced logos. I'll talk about that in a moment. And then we have this intron region, the whole intron region, and then we have acceptor region that ends up with AG. So you can see the big proportion of AG, and then next exon it starts. What are sequence logos? Sequence logos are visual representation of a position specific distribution. So at particular position, what is the frequency of some nucleotide? If the frequency is more the frequency is, more big it will be. So the height or the size of that particular nucleotide gives a idea about a relative frequency with respect to the other nucleotides. So it's easy for nucleotides, but we need colors to depict 20 amino acids. So sequence logos are good representations as far as the DNA nucleotides are concerned. And proportions of each nucleotide are in relation to their observed frequency. So sometimes we have more than one of the the heavy frequency nucleotides. So we can order them and put them, the highest one at the top and the rest of them at the bottom in a hierarchy. Non-canonical splice sites can be GCAG, which is 0.69%. It can also be AT and AC, and there are some others. So we can get the information contact. We can see the observed frequencies of some nucleotides around those these donor and acceptor splice sites, and we can use those frequencies in order to calculate the relative frequencies of those particular nucleotides, and then we can get a scoring function out of it. That's what we did in TSS, and we can use those scoring function in order to have an estimate that whether those particular sequences have a strong evidence to have those these splice junctions. So here is the acceptor site distributions. So if we look into the acceptor sites, we say that position number -2 and -1 are the places where this acceptor site is there. So as we see, the frequencies over here are 100%. It's A and it's G over here. So AG is predicted. But if we look into the neighbors at position -3, there are mostly C's, and at position number -4, it may be C, or it may be TU. In case of DNA, it's T, or in case of RNA, if you are comparing, it's U. So they both of them, they are comparable, but C is mostly present. We look into -5. We see the proportion of T and U is higher than we have C's. So same way as we did in the TSS, we can have those frequency distributions. Look into the donor sites. We have G's here and we have T's here, or U's here. We label them as 1 and 2, and then at different positions which are neighboring to them. So in this way we can open up our window. We can include like 10 or 15 different neighboring positions Here are the dominant ones. So we can come up with a model. So here we can plot the frequencies of those nucleotides, and we can score the segment as a candidate, donor, or whatever acceptor site by using this relation, where we can take their frequencies from these frequency tables, and we divide that with their expected frequency, and then we take a log score, and stronger the values, stronger is the evidence that we have predicted a donor or acceptor site. So like TSS, the flanks of splice junctions, they also show the bias distributions of nucleotides, and these distributions can be used for their prediction.